In what ways are plants like people? In 1975, Jerry Baker published a book called Plants Are Like People. It's a gardening book that is very cute in terms of comparing people and plants. For example, what temperature of water do plants like? They like the same temperature you would like. How much light would they like? They like about the same amount we do, with individual variations. We enjoy a sunny day, and so do plants. They have a circadian rhythm of rest and activity, and so do we. We drink water, they need water too. And there's more. A very hot topic of research is plant intelligence. Yes, plant intelligence. This topic is a bit controversial because there was a book called The Secret Life of Plants, first published in 1989. Definitely written for the lay enthusiast. The book has chapters entitled Plants Can Read Your Mind and The Mystery of Plant and Human Auras. This was the book that claimed certain types of music encouraged plant growth. Surely this must be one of the most often repeated elementary school science fair experiments of all time. But it's never been confirmed in a repeatable lab study. Even if plants are responsive to sound, it's the people taking care of plants who are more likely to be affected by human music. I think most botanists thought the book was kooky in a new age sort of way. However, that book was a wild success, still ranking number 20 on Amazon's top-selling botany books almost 3 decades after it came out. And in some ways, the book created a rift in botany that persists to this day. Scientists interested in the ideas of plant communication or plant behavior tend to be labeled on the fringe by more mainstream botanists. But there are also attempts to rethink plant behavior from a more mainstream perspective. An Italian botanist named Monica Gagliano is one scientist studying plant communication. With colleagues, she published a 2012 paper entitled Towards Understanding Plant Bioacoustics. This is not a paper about the sounds that plants make. By the way, if a tree falls in the forest, does anyone hear it? Okay, it's not that kind of paper. The authors state, here we present a rationale as to why the perception of sound and vibrations is likely to have also evolved in plants. The perception of sound, these authors are investigating what plants hear. It's pretty convincing stuff. They argue that many animals lack eardrums, yet they can still hear. Snakes hear through their jaws, and insects hear through their antennae. Why, they argue, could plants not hear through some organ or mechanism we don't yet understand? They make a good argument that there are constant sounds in substrates such as soil. Animals who live underground still hear, though their hearing is generally reduced. The authors outline an experiment where the roots of young corn are shown growing towards a sound emitted in water. Pretty much everyone knows that roots need water, so it would make sense that roots will grow towards sources of water. It's even been reported that roots will grow towards pipes with water in them, even if the pipes are sealed and not leaking any water. This also suggests that the roots are somehow sensing the sound. This same botanist, Dr. Monica Gagliano, has been experimenting with Mimosa pudica to test its memory. After all, it's the sensitive plant. It's a small shrub-like herb, something botanists call frutescent, which means shrub-like. This plant is native to the tropics of North and South America. There are about 430 species of mimosa, but only a few species display the special trait that Mimosa pudica displays. Its remarkable trait is when touched, its leaves collapse immediately. In fact, the scientific name pudica is Latin for shy. Before I tell you about the work with mimosa's intelligence, let's explore the mechanism of this leaf movement. Most plants don't move like this, and in fact, many people think that plants don't move at all. It's true that the mimosa plant itself doesn't move, but the movement of the leaves is rather instant. How do the leaves move so fast without muscles? In order to understand this, we need to go over a basic biologic principle called osmosis. Now, you've probably heard people say something like, "I'll learn it by osmosis." And this really irritates me because osmosis is only possible when there's a movement of water. They're actually thinking about a more general process of diffusion, which is the movement of anything from a high concentration to a low concentration. So if I bring a cup of coffee into your room, you're likely to smell it before too long because the scent molecules of coffee are moving from a high concentration, which is just above the coffee, to a low concentration, which is everywhere else in the room. So when someone says they'll pick it up by osmosis, they mean they'll pick it up by diffusion from a high concentration of knowledge to a low concentration of knowledge, which would be their brains. So osmosis is a special case involving the diffusion of water. This process will also be important when we think about movement of water through the plant. So in osmosis, water will move from an area of high water concentration to an area of low water concentration. Now, you're probably wondering, how can you have low water concentration? You have water or you don't. Well, that's not exactly true. Water has more water in it than, say, Kool-Aid. In Kool-Aid, there are other things in the water, and we call them solutes, sugar, flavorings, color. So if water moves from a high concentration to a low concentration, think about which way the water will move if we have two chambers separated by a membrane that is only permeable to water. In one of the chambers is Kool-Aid, and in the other chamber is pure water. Remember, the membrane separating the chambers is only permeable to water, and water, like everything else, will move from a high concentration to a low concentration. Which way will the water move? If you said the water will move from the water side into the Kool-Aid side, you're right. The concentration of water was greater in the pure water than the concentration of water in the Kool-Aid. Now, what does this have to do with plants? The membrane that is permeable only to water in our example is much like a plant's membrane or cell wall. So back to our mimosa example. The mimosa also has a special thickening at the base of the leaf called a pulvinus. These cells are what control the drooping of the leaves. Now, remember back to our osmosis example. Consider that the leaf is fully turgid. That's a botanical term that is the opposite of wilting. So the leaves are flat, but when you touch them, they droop or wilt immediately. When touched, the plant produces an electrical signal that causes potassium channels in the cell wall of the pulvinus to all open. When all the potassium rushes out of the cell, the water will follow it by osmosis, thus causing the pulvini to wilt or become flaccid, which makes the leaves droop. A flaccid cell also causes plasmolysis, which is when the cell shrinks due to lack of water, and the cell membrane pulls away from the cell wall. A flaccid cell doesn't always have plasmolysis, but if it remains flaccid long enough, the plasmolysis will occur. Now, back to our Italian scientists looking for plant intelligence in mimosa. As we just learned, this plant will respond to touch by drooping its leaves. It would probably do this as a response to herbivory, a trick as if to say, I'm not a good plant to eat. So in this study, Dr. Gagliano is testing the memory of the plants. Yes, their memory. How well can the mimosa plant remember something that happens to it? Her experiment went something like this. She would drop the mimosa plants and the leaves would collapse, just as we discussed. She would repeat this several times until the mimosa stopped collapsing their leaves. Okay, the potassium channels can't reboot or some sort of physiological argument. But then she would drop the plants all week, a whole week later, and the leaves would not collapse. To test them, she would give them some other stimulus, and the leaves would droop immediately because they had not been conditioned for a response. So if plants can respond to sound, respond to touch, and perhaps even have memory. How else are plants like people? And in what ways might they be different? We can build this discussion from the bottom up by thinking about plant and animal cells. There are a lot of ways that the cells of plants and animals are similar, but there are a few important ways that they're different. Let's look at the differences first, and then outline some of the similarities. Plant cells have three important structures that animal cells don't have. Plants have number 1, a cell wall, number 2, a vacuole, and number 3, plants have chloroplasts. Let's start with the cell wall. All cells, including animal cells, have some sort of boundary between the cell and everything else, the extracellular fluid or matrix. Both plants and animals have a cell membrane, but plants also have a cell wall. This is just the way it sounds. The cell wall is tougher, usually thicker, and has more structure than a cell membrane. In plants, the cell membrane is called the plasma membrane, and it's inside the cell wall. Put another way, on top of the plasma membrane is the primary cell wall. And the cell wall is made up of polysaccharides, which is a fancy word for a lot of sugars all stuck together. One of these polysaccharides is cellulose. We can't digest cellulose even though it's just a chain of glucose molecules. Instead, cellulose is a component of dietary fiber. Cellulose is also the most abundant macromolecule on Earth. These cellulose molecules are long strings of glucose that crisscross each other to add structure and strength. We call the cellulose molecules microfibrils, and embedded around these are pectin molecules. Pectin is the primary component in jams that gives them their viscosity. And the same here with the cell wall. The pectin provides flexibility. Primarily, the cell wall keeps the cell from bursting under turgor pressure. Most plant cells are turgid or full of water to maintain their shape. An animal cell would burst under that kind of pressure. But plants are able to get such high pressures in their cells because the cell wall holds in the water. Plant cell walls also have small pores in them, which allow for water to pass into the cell and a few small nutrients. Remember, earlier in this lecture, we talked about osmosis as being the movement of water through a semi-permeable membrane. The cell wall, even though it's a wall, is an example of a semi-permeable membrane. Cell walls can be either primary cell walls, which are still growing and living, or they can be secondary cell walls, which are mostly no longer alive, but contain a substance called lignin to provide structure. In animal cells, the cell membrane is important because it receives many signals from the rest of the body. Let's think of the cell membrane as a cell phone. Get it? Cell phone? It's sending and receiving signals. Okay, seriously though, the cell membrane contains receptors on the surface of the cell membrane that receive signals from the rest of the body. Let's say the cell needs some glucose. It has a glucose receptor on the cell membrane that is open, and so it receives glucose. The evidence that plant cell walls regulate responses in the same way is just not that strong yet, and this is a big interest in botanical research right now. The second unique component for non-woody plant cells is the vacuole. The vacuole is an organelle, which is sort of the way it sounds, a cellular organ, or an organ for the cell. That's an organelle. The vacuole's main purpose is to store water. When you look at a cartoon of a plant cell, about 90% of that cell is taken up by the vacuole, and everything else is sort of squeezed around that organelle. The vacuole stores water, but this water, as we've mentioned, is also the primary component giving cells their structure. Both vacuoles and cell walls can be used to clean up toxic waste in a process called phytoremediation. Phyto is Latin for plant, and remediation means to restore. For example, the mining industry sometimes leaves residual heavy metals in soils and waterways. Certain plants called hyperaccumulators can draw heavy metals up through their roots and store these metals in their tissues. For example, rapeseed, or Brassica napus, is a plant in the mustard family that gives us rapeseed oil, which is also canola oil. This brassica plant is also a good accumulator of metals like mercury, lead, and chromium. Generally, the heavy metals can be stored in the cell walls or in the vacuole of the plant. The third unique component that plants possess is the chloroplast. The chloroplast is an organelle which, as we said, is a mini organ in the cell. Chloroplasts contain chlorophyll, and they are the main site of photosynthesis. In some ways, chloroplasts resemble the mitochondria of an animal cell. The mitochondria is the main site of energy production in the animal cell. In fact, the chemical reactions are exactly opposite in the chloroplast and the mitochondria. The chloroplast takes in sunlight, carbon dioxide, and water and creates glucose and oxygen and energy. The mitochondria takes in glucose and oxygen and creates carbon dioxide and water, a process known as cellular respiration. All plant cells also have mitochondria. Why? Like animals, just to break down the glucose produced during photosynthesis. But mitochondria are especially important for cells that don't photosynthesize, like the roots. It also implies that these cells need oxygen. This is one reason we aerate our lawns, so that the roots of the grass can get oxygen. Both the mitochondria and chloroplasts are thought to have originated from free-living bacteria that became parasitic or somehow lived symbiotically with other cells. Over time, these bacteria changed to become mitochondria of modern plant, fungal, and animal cells, while others became the chloroplasts of modern plant cells. This process of bacteria becoming symbiotic organisms living inside other cells is called the theory of endosymbiosis. Both plant cells and animal cells contain a nucleus. The nucleus contains the DNA, or deoxyribonucleic acid. The genetic information, or genes, are contained within the DNA. Perhaps unsurprisingly, DNA works pretty much the same in plants as in animals. This is unsurprising because this code was developed very early on in evolutionary history, so it has been retained over many millions of years. In order to understand how DNA and genes work, it helps to understand the structure. Deoxyribonucleic acid is a chain of nucleotides. A nucleotide consists of a 5-carbon sugar, that is the deoxyribose, a phosphate group, and a nitrogen-containing base. This last part, the nitrogen-containing base, can either be adenine, thymine, guanine, or cytosine. These get abbreviated A, T, G, and C. Movie buffs may remember the science fiction film Gattaca. Note that the title uses only A, T, G, and C, which is the genetic code of DNA. When I first learned about the genetic code, it seemed sort of unimpressive that the entire blueprint for life came down to these 4 nucleotide bases. But it does. It's helpful to think of it this way. The entirety of the English language comes down to 26 letters. The similarity is they each form words. The 4 nitrogen-containing bases form codons, and these are sort of like words. Each 3 nitrogen bases forms a codon. That codon will code for a particular amino acid. Now, here's some math review. If there are 4 bases and 3 bases per codon, how many codons are possible? That would be 4 to the 3rd power, or 64. Interestingly, as I said, the codons will code for an amino acid, but there are only 21 amino acids essential for life. So all our essential amino acids might have been possible with only 3 bases, except for one thing: the complementarity of DNA would be lost. Complementarity means that the nitrogen bases bind to each other, and that's how they create the double helix we all know and love. A binds to T, and C binds to G. The way proteins are made in plants and animals is the same. It's due to the complementarity of the DNA. To make a protein, first the DNA is read by messenger RNA in the nucleus. Second, the messenger RNA takes the message out of the nucleus to the ribosomes, where it's translated by transfer RNA. Third, The transfer RNA then assembles the amino acids in the sequence that is dictated by the messenger RNA. There's a special codon called a stop codon to tell the transfer RNA that the protein is complete. No more amino acids need to be added. The amino acids strung together make up a protein. This process of creating proteins from the recipe in the genes is happening in the cells all the time. Whether or not a gene is being activated is known as gene regulation. So the DNA contains the genes, the genetic information from the parents. Humans are diploid organisms, which means that they have 2 copies of all the DNA, one from mom and one from dad. Sometimes these 2 copies have the same information for a certain gene, and sometimes they have different information for a certain gene. That bit of information for a certain gene is called an allele. I like to think of alleles as flavors. If the gene is ice cream, then the flavors are chocolate, vanilla, strawberry. There might be many alleles in the population, but any diploid organism will just have 2 flavors of alleles, one from mom and one from dad. Most animals are diploid, and many plants are too. But plants can also have more copies, which is one way we get different varieties of plants. Your apple, for example, might be diploid, triploid, or tetraploid, depending on the variety of apple. Plants and animals not only share the same DNA building blocks, they share many of the same DNA sequences or genes. For example, the human genome, or collection of all the human genes, and the rice genome share about 25% of the same genes. However, when hearing such a statement, it's also important to realize that shared genes are expressed via alleles that are very different for humans compared with plants. On the other hand, some proteins, such as cytochrome C, which is important for respiration, are found in all plants and animals. We owe a lot of basic information about genetics to a monk living in the 19th century. Gregor Mendel did his classic pea experiments in what is now the Czech Republic during the 1850s. He actually developed these ideas about inheritance without knowing anything about genes, alleles, or DNA. I love this example because I think it demonstrates how science is a useful construct, even when there isn't that much that's understood. In Mendel's time, people thought inheritance was all blending. If your mom had blonde hair and your dad had dark brown hair, you would have lightish hair. Sometimes that's true, but a lot of times it isn't. By using pea plants as a model organism, Mendel was able to meticulously breed pea plants and make observations about the outcomes. They have male and female parts, so they can be self-fertilized within the plant, or they can be cross-fertilized, that is, between different plants. Mendel did his crosses, or pollination, with a paintbrush. He would get pollen from one flower and then put it on the pistil of another flower. He would use the pollen and pistil from the same flower if he wanted a self-pollination cross. He then had to wait for the fruit to form before he could tell what he got. Pea plants take about 3 weeks to form fruit after the plant flowers. This is one reason why botanists don't use pea plants as model organisms much anymore. But for Mendel, they worked well because there are a number of visible differences called phenotypes that can be observed from various crosses. The 7 traits that Mendel observed were seed color, seed shape, seed coat color, pod shape, flower position, and stem length. So even though Mendel didn't know it at the time, the 7 traits that he looked at were 7 different genes. The physical trait of the gene was the allele. So looking at seed shape as the gene, plants could have a smooth allele or a wrinkly allele. So to determine if blending was occurring, Mendel crossed 2 homozygous parents. Homozygous means that a given parent had the same 2 alleles. So the wrinkly parent had 2 wrinkly alleles and the smooth parent had 2 smooth alleles. When he did this, he found that all of the offspring were smooth. He had disproven the idea of universal blending because none of the seeds were a blend of smooth and wrinkly. He also observed that one allele, smooth, was dominant over the other. He then wanted to know if these traits could be inherited independently. So he did another cross of two parents that were both heterozygous for the wrinkly trait, which means that each parent had one wrinkly allele and one non-wrinkly allele. In genetics, the dominant allele is usually written as a capital letter and the recessive allele is written in lowercase. He was able to show that the allele for the wrinkly could be inherited separately from the allele for color, so that there were all sorts of combinations that were possible. Green smooth, green wrinkly, yellow smooth, and yellow wrinkly. Even though there were only 4 phenotypes that resulted, there were 10 different genotypes. Thus, Mendel was able to show some pretty important features of genetics. And these basic features are true for plants and people. First, he was able to show that blending doesn't always occur, and this is because the alleles segregate from their parents. This happens when parents form gametes, or egg and sperm. The egg and sperm only get one copy of an allele, and these segregate during gamete formation. Mendel's second finding was the idea of independent assortment, which means that genes don't have to be inherited together. Mendel's third observation was that of dominance, meaning that some alleles are dominant over others. Some textbooks call these Mendel's laws, but this is inaccurate, I believe. Certainly segregation occurs during gamete production, but some genes are in fact linked and will be inherited together. Also, some alleles are codominant, meaning that both alleles can be expressed. An example of codominance is seen in the human blood type AB. This blood type means that an A allele from one parent and a B allele from another parent would both show up in the blood phenotype. An example in plants would be a pink carnation, which would result from the breeding of a red carnation with a white carnation. Both colors get expressed, and pink is the result. So Mendel's ideas are good models for how much of genetics works, but it's not how genetics works all the time. So we know that the basic genetic principles are the same in plants and animals, but we learned that there are 3 unique features of plant cells: the cell wall, the vacuole, and the chloroplast. A group of cells together makes up a tissue. Because plants have such different cell types from animals, they also have different tissue types than animals. There are 3 main tissue types in plants, and these are categorized by the thickness of the cell wall. Tissue with the thinnest wall is called parenchyma. Thinking about form and function, where might plants want to have the thinnest cell walls? Certainly, somewhere there might be a lot of exchange. The leaves. And parenchyma is the tissue that makes up much of the internal structure of the leaves. Collenchyma is a tissue with irregularly thickened cell walls. These are structural cells, though they are not found in the roots. Typically, they are most common in the stems of herbaceous or non-woody plants, like celery. Sclerenchyma is tissue that contains cells with thickened secondary walls that may contain lignin, which is a substance that provides thickness. Sclerenchyma serves a protective or supportive function in the plant. This tissue type is easy to remember when one thinks of arterial sclerosis, which means a hardening of the arteries. Sclero comes from the Greek skleros, meaning hard. Anyone who's eaten a pear has intimate knowledge of sclerenchyma because pears contain a special cell called stone cells that have lots of sclerids. The grainy or gritty taste in pear flesh comes from the sclerids. In fact, under a microscope, these cells are so recognizable, they've been used to solve crimes. Chromoplasts from red bell pepper are another example. Forensic botany is the use of botanical material to solve crimes. Dr. Jane Bach and colleagues first published a lab manual about this for the U.S. Department of Justice in 1969 with information about plant tissue types and plant cell walls that have been used to solve dozens of cases. Sclerids of pears are so recognizable that one can still find them in the gastric contents of victims. It turns out that there are a number of recognizable cells in plants that have been used to detect where and when last meals were eaten. So Plant intelligence may be controversial, but there's no doubt that plants can solve crimes. Plants and people definitely have some genes in common and use some of the same building blocks of DNA to make proteins. Like people, plants have tissues, but plant tissue types depend on the thickness of the cell walls, which our animal tissue types don't have at all. So the form and function of plant tissues is different. Most important and most surprising, given these differences, plants are able to sense their environments and respond. The open question is whether plant sensation is akin to sensation in animals, and whether plant responses to their environment are akin to animal behavior and communication.